Hello everyone, and welcome to this audio podcast. This audio podcast overview of this of a research poster presentation for the remit of UKKW 2025, the UK Kidney Week 2025. The research poster presentation this research poster presentation, some collaborative works on behalf of the Renal Patient Support Group, the RPSG, looks at an exposition on stratified receptors, the idiosyncrasy surrounding early transplant immunobiology, a comparative perspective. In this research poster presentation, sort of overview, there will be a look at an introduction, a look at chemokine receptors, and leukocyte trafficking in human AAR. A look at immunology and stratifying receptors, a look at sort of the aims and objectives and some methodology, and then a look at transplant immunobiology transplant immunobiology and idiosyncrasy, and then look at immunosuppression therapies and AAR, and then a look at sort of a brief look at bridging healthcare disparities and inequalities, and then a conclusion, and then a sort of a look at some main points as part of this research poster presentation overview. So to begin with some introduction. So chemokines chemotactic cytokines are a large family of of small but structurally related proteins that intervene a wide range of biological activities. As part of normal immunology or immunobiology, chemokines are a critical component of leukocyte trafficking, essential for immune system architecture development and immune surveillance. Chemokines binding to receptors on leukocytes mediate the behind the scenes plays between antigen presenting cells and host T cells in lymphoid tissues that can acutely sort of destroy an allograft and prompt an inflammatory inflammatory responses. So now a look at a sort of chemokine receptors and leukocyte trafficking in human AAR. So these are mediated. So chemokines or chemokine receptors are sort of mediated through a family of G coupled, a family of G protein coupled receptors with several transmembrane across 7 transmembrane domains, and the nomenclature used to describe these receptors is based on the classes of chemokine ligands that interact with the receptor So, for example, C and then CC and then CXC and CX3C receptors. Many chemokines bind to several different receptors, yet the differential expression of receptors by distinct leukocyte subsets is an important component for specificity of chemokine action. The complexity of apparent redundancy of the immune system is thought to provide a high degree of effectiveness and flexibility in vivo. Investigations with knockout laboratory mice or murine models, especially when teamed with the use of in wild type mice or of blocking monoclonal antibodies or MAB to a corresponding receptor, have provided initial laboratory analysis as to what all the complexity of chemokine biology might mean to the highly practical in vivo world of transplantation. Immunobiology and stratifying, now looking at sort of immunobiology and stratifying receptors. So for CXCR3, virtually all T cells are at sites of non-renal human inflammation with prominent mononuclear self-infiltrates are receptor positive. Growing evidence suggests that differential chemokine receptors receptor expression may be crucial for the generation of a T helper type 1 or T helper type 2 type immune response or responses because specific chemokine receptor expressions have been shown to characterize these T helper cell subtypes. T helper type 1 cells also appear to preferentially express the chemokine receptors CXCR3 and CCR5 when T helper type 2 cells display CCR4, CCR8, and some CCR3. The chemokine receptors expressed by different populations of T cells may thus dictate to a significant degree the tissue infiltration of T helper type 1 and T helper type 2 cells and the direction of the eventual immune response. Now look at sort of the aims and objectives as part of this research poster presentation overview. So the review looks at an exposition on stratified receptors and the idiosyncrasy surrounding early transplant immunobiology. Understanding of cytokines and chemokine receptors can prompt better direction of immunosuppression therapy or IST and maintenance therapy regimens or protocols in the renal allografted cohorts. This review or this overview looks at an understanding surrounding donor-specific onset of onset of acute rejection episodes. So in as part of the, in addition to the aims and objectives, some methodology, although the publication isn't a, the publication or the research poster presentation overview is not in regard to sort of informing of laboratory data. However, is a publication, a research poster presentation in looking at sort of an interlink around specific immunobiological components and their basis for and part of immune responses. So the methodology is where the aims and objectives are additionally met by investigating early literature, highlighting importance of bridging healthcare disparities and inequalities through patient and public involvement or PPI, and summarized workings of social media platforms, the RPSG and CADARs, and additionally the value of an effective multidisciplinary team and healthcare workforce. So now looking at transplant immunobiology and idiosyncrasy. The varied expression of chemokines within the allograft brings into question the best approach to targeting chemokines for IST pharmacotherapy. It would be better to block early innate responses to allograft, thus preventing the infiltration of non-allo AG or specific cells and inhibiting the adaptive immune response. A few questions that have been asked in earlier works was which chemokines are most crucial for graft infiltration, and must all chemokines be blocked, or is neutralization of only a specific few sufficient to prompt allograft prognosis or survival. So further, as observed in cardiac cells and gallium nitrate treated and gallium treated gallium nitrate treated investigate gallium nitrate treated laboratory investigations, or through those investigations, some chemokines persist even after graft allograft acceptance, with no apparent determinant to no apparent detriment to allograft. CXCL10 knockout murine mice using CXCL10 donor cells did not express but exhibited a prolonged allograft prognosis or survival. This suggests that the ability of allograft to secrete T cells recruiting inflammatory chemokines may directly result in self-acute allograft rejection, or AAR. Now a look at immunosuppression therapies and AAR. So IST surrounding transplantation has undergone more incredible advances over the last 3 decades than perhaps any time in history. It is now possible to be more selective surrounding IST pharmacotherapy, and sometimes irrespective of human leukocyte antigen, or HLA, serology and tissue typing investigations. However, it is an ongoing process that incorporates the use of IST and laboratory advances, thus understanding different points in the activation cascade of CD4 positive and T helper cells, Th cells, and the reaction of CD8 positive T cells. It may also be possible to manipulate the immune system, that the organ specific immune response may be switched off or rendered sort of tolerant, thus removing the need for using ISTs. Since the mid-90s or the mid-noughties, there had been a burst in the quantity of new pharmacotherapeutics available amongst solid organ transplantation specialties. And now a look at bridging healthcare disparities and inequalities. So co-developing in keeping with PPI, or patient and public involvement, patients and professionals working together has now become more important, so thus to alleviate the technical burden of frontline professionals, but also to encourage decision makers to think differently about the way healthcare could be, the way healthcare could better clinical practices. Collaborations are important in developing more inclusive practices in bridging educational services and transplantation. So, for example, healthcare service users may find it advantageous having one-to-one consultations with an AHP or allied health professional and or healthcare scientists surrounding renal educational themes, thus enhancing patient access to care. The RPSG and KDOS social media platforms have looked to follow up on this through collaborative research. So in conclusion, as part of the conclusions then, investigation in the role of chemokines and surrounding AAR research, acute allograft rejection research is still challenging, primarily because healthcare professionals that could offer educational support through integrated PPI-centric services and MDT involvement for healthcare service users have not been piloted. PPI to help understand where healthcare service improvement enhancements enhancements are needed are needed and are still sort of important or pertinent when it comes to integration and joined up thinking. Research surrounding the RPSG and KDARS platforms have been propagating integrative practices involving patient and health professionals through collaborative efforts. So now look now in on the main points. The main point is as part of the main points, three aspects. Initially, what is known, and then what this work adds, and then the potential impact. So, what is known? Laboratory investigations have shown that expression of multiple chemokine receptors allow navigation by leukocytes systemically laboratory investigations have shown that expression of multiple chemokine receptors allow navigation by leukocytes systemically through a variety of multiple chemokines. This then allows different chemokine receptor pairs for activations, transmigration, and retention, respectively. The varied expression of chemokines within the allograft brings into question the best approach through for IST pharmacotherapy. And as part of what this study adds, or this investigation adds, patients suspect or high risk for confirmed CKD not only need access to laboratory investigations surrounding transplantation, but additionally to understand aspects pertaining to immunobiology through themed educational services aligned to PPI initiatives. And as part of the potential impact, research and evidence-based practices surrounding the RPSG and KDARS platforms have been encouraging integrated practices since 2009 and 2014. That ends this research poster presentation overview on an exposition on stratified receptors, the idiosyncrasy surrounding early transplant immunobiology, a comparative perspective where this research poster presentation is on behalf of the RPSG.